Hello, welcome to BioBrainiacs. Today we'll build the background knowledge you need before starting the experiment. We'll learn what bacteria are, how culturing works, what you'll do on Monday, and what we'll observe on Thursday. So what are bacteria? Bacteria are microscopic single-celled organisms found almost everywhere around us. When they multiply enough, they form visible colonies that we can observe in our lab. Where are bacteria found? Bacteria exist on almost every surface you touch, even ones that look clean. That's why swabbing different objects will give each of you unique results. Why grow bacteria in a lab? Growing bacteria in a lab lets us actually see something that's normally invisible. It helps us compare surfaces and understand the microbial world around us. What does culturing mean? Culturing means growing microorganisms in a controlled environment so we can observe them. Our experiment uses the same method to make bacteria visible. How do bacteria grow? Bacteria grow by dividing into identical copies, sometimes very quickly. Over time, millions of cells build up to form visible colonies. What do bacteria need to grow? Bacteria need nutrients, moisture, and time to grow. Agar plates provide the perfect surface and nutrients for colonies to form. What is agar? Agar is a gel-like material that provides nutrients for bacteria. It allows bacteria to grow into colonies we can easily see. Petri dishes explain. Petri dishes hold the agar and keep bacteria contained. They make it easy for us to observe growth safely later. What will you do on Monday? On Monday, you'll receive a pre-made agar plate and swab any surface you choose. Then you'll spread the swab across the agar, close the plate, and return it back to me. Choosing with the swab. You can swab almost any school-appropriate surface that interests you. Choosing different objects will make comparing results more interesting. How do you swab the agar plate? Gently move the swab across the agar surface for about only 10 seconds, then close the plate immediately so bacteria can grow safely inside. What's there to do after you finish on Monday? So after you return your plate, it will be stored safely and left undisturbed, and over the next few days, bacteria will grow into visible colonies. What happens while we wait? So during the week, bacteria multiply and form colonies on the agar, and by Thursday, these colonies should be clearly visible. Thursday, observing results. On Thursday, we'll reunite and observe your plates without opening them. We'll compare results and look for patterns across the group. So what is this colony term that we keep speaking of? A colony is a visible cluster of many bacteria growing together. Most colonies start from a single bacterium that multiplies over time. Colony differences. Each plate will look different depending on what was swabbed, and colonies can vary in size, color, and shape. Comparing surfaces. We'll compare which surfaces had more or less growth, and this helps us think like scientists and look for patterns. Why some surfaces have more bacteria? Surfaces that are touched often usually have more bacteria. Environment and cleaning habits can also affect growth. Variables in our experiment. The main variable in this experiment is the surface you choose to swab. Keeping everything else the same helps us compare results fairly. Real science connection. Real scientists use similar methods to study bacteria in hospitals, labs, and the environment. Our experiment reflects the same core microbiology techniques. Why don't we open the plates later? Once bacteria grow, we keep the plates sealed for safety and accuracy. We'll observe everything through the lid instead. Key takeaways. Bacteria are everywhere and grow when given nutrients and time. This experiment helps us visualize a normally invisible world. Key reminders for Monday. Use one swab and one plate, cover the agar for about only 10 seconds, then close it immediately, and return Thursday ready to see and compare your results. Thank you, and see you Monday.